talk with showing, let's see if I can do this. Sorry, that's okay. Got it. Showing our research participants, they've all given permission. This is Fragile X. These are our families. These are people who become friends. These are our research participants. And so whenever I'm presenting data, yes, they are a dot on a screen a lot, but these are the people that are coming into the lab, and it's really meaningful. So always want to bring it back to that. I know a lot of you know about Fragile X here, but just to get us all on the same page, it is the most common inherited monogenic form of intellectual disability and cause of autism spectrum disorder. In addition to that intellectual disability, the primary phenotypes are inattention, hyperactivity, and irritability. Fragile X is caused by that CGG repeat expansion on the FMR1 gene and the X chromosome, and that's disrupting that protein, FMRP. We'll be talking about FMRP a lot today. And this is why, because it's critical for that brain function, it's critical for synapses, and we think it's related to the clinical phenotype. There's some early data, even from 20 years ago, that was suggesting that the amount of protein expression was related to IQ, but with some of our nice new optimized assays, we're even able to continue to show that. So as Matt already introduced part of my talk, which was great, is that despite being a single gene disorder with a really well-established animal model, the FMR1 knockout mice, we're still at this kind of crossroads where we've had a lot of beautiful preclinical data that basically solves and cures Fragile X. There is a father that Craig always refers to of it's a great time to be a Fragile X mouse in the world because we cure Fragile X almost every time in the mouse model. But right now, there's no FDA-approved treatments for Fragile X. Those preclinical trials have failed to translate into human studies. And a lot of those pharmacological interventions that we are using that are having symptomatic impact on irritability or hyperexcitability in Fragile X, nothing is really targeting some of those core features of intellectual disability and other cognitive. So again, we're kind of at this, what are we going to do next phase? And so it's important to really think about what is causing Fragile X. And so we have our FMR1 gene and this repeat expansion. And so in typically developing individuals, there's CGG repeat expansion under 45. There's the premutation. I'm not going to go too much into the premutation. It's very complicated, and Matt is doing some really nice work there. And then the full mutation is that CGG repeat expansion over 200. Whenever you have these over 200 repeat expansions, you're going to have methylation, which basically causes more or less the gene to turn off in terms of stopping mRNA production, which is then necessary for FMRP production. And so you kind of have this cascading effect that you're depleting mRNA, you're depleting FMRP, and with FMRP is really important for synapses and brain development. That's going to have a cascading effect.
Okay, how are you?
So because it's on the X chromosome, things look a little bit different for males and females. So if we're thinking about the fully methylated full protein males, these are individuals who more or less have no FMRP production. We will talk about that a little bit later though. And then in mosaicism, you can have either methylation mosaicism, so they're not 100% methylation, there is some weakly methylation, or you could have repeat mosaicism, which is in that CGG repeat count. And so you're still seeing that some of that FMRP is expressed in some of those cells versus not. And then in females, again, it's a little bit more complicated of a picture where sometimes there's going to be FMRP expression and sometimes there's not going to be due to X inactivation. Our lab, led by Nina Gross and then Anna Boggs, who is currently in med school, optimized our Luminex-based immunoassay for protein production. And so there's been a lot of different methods to kind of analyze and quantify FMRP. But what's really unique about ours is that it is highly, highly sensitive to low numbers. And this is what is something very important that we think hopefully will move the field forward. And so in our recently published paper, we had a decent amount, about over 100 individuals Fragile X, 49 premutation carriers, and 35 typically developing individuals across a wide range of age. And we're kind of showing exactly what you would expect, that individuals with Fragile X have less protein than pre-mutation carriers, and typically developing individuals. However, if we start zooming in to these individuals with Fragile X, what we're seeing is that, of course, females are expressing more protein than in males, as we would expect given X inactivation. But there is a good amount of these males with Fragile X that are expressing trace levels of protein. So zooming in further, we're seeing that in full mutation, Fragile X, about 40 to 50% of full mutation, fully methylated males, based on their Southern blot and PCR methods, are expressing trace levels of protein. This is a pretty novel and new finding. We just sort of put them all in a box together, assumed that, you know, all males, fully methylated males, were expressing zero protein, but there's actually trace protein there. So that's novel in itself, but does it mean anything? And so we'll go into that a little bit later. The other important thing about our assay is that, at least in adolescents and adults right now, we know that it is stable over time. So these are a bunch of individuals who participated in a drug study. And so over the course of 13 plus weeks, we're seeing that their FMRP levels are relatively the same. So if they're in that trace level, they're going to stay in that trace level. If they're in that zero, they're still expressing zero protein. And this is meaningful so that we can know that if we're collecting protein at time point A, and a year or two from now, we can still probably identify where they're going to be. We haven't analyzed a bunch of our younger individual data. We have a lot of longitudinal data from infancy to 10 years old that we're hopefully getting into now in the near future to determine whether FMRP levels are changing over time. So sorry, this slide has the 30 to 40% that's showing expression based upon, as we're getting more data into it does look closer to 40 to 50% now. But real quick, so if we're showing that there is that FMRP expression, we would then kind of think biologically that there's probably some mRNA expression too. It doesn't just come out of nowhere. And so after we optimized our FMRP assay, we worked on our mRNA assay. Again, got to thank the lab for all the things that they do. We recently published this paper. And the bottom line is that, again, individuals with full mutation, fully methylated males are expressing some mRNA as well. But there is a subset of individuals down here that do have mRNA, but it's not getting translated into protein. We don't know why yet. And so this is something we're definitely trying to figure out. We have new advanced technology to really try to figure out, is it based on CGG repeat count numbers? Is it based on those methylation rates? Whenever we're looking at Southern blots and our PCR methods, we're sending those to Rush at Liz Berry-Kravitz. They're sending them back. And we're just getting a report that says over 200 CGG repeats or fully methylated. But there's probably a lot of subtlety in there as well. It's probably a rounding error, but no one can really tell us on that side of things where it is. And so our advanced nanopore technology hopefully will be able to share some of that as well. And so, like I said, there's this evidence of an incomplete silencing of that FMR1. And so we do see that a subset of those males with FMR1 expression, but not all of that translates into protein. And so this is sort of, I talked about these paradigm shifting things. So the first paradigm shift is really that there is a subset of what we're considering fully methylated, full mutation males that express this trace level of protein. So this is biologically different than we ever thought about Fragile X. We just kind of, again, put them all in one category. These individuals are biologically different than we knew before. Importantly, not all of those individuals are expressing. And again, those traditional methods that we're using to diagnose Fragile X may not be sufficient or sensitive enough to really determine that FM status or full mutation, fully methylated status. And so we need some more better improved molecular technology to really tease apart that molecular variation. And like I said, we're hopefully moving in this direction. We just got training a couple weeks ago using our new technology. So stay tuned on kind of that molecular front of things. But if the biology is different, does that mean the clinical presentation is different? Is it actually meaningful that we found this 40 to 50% of individuals that are expressing protein in FMRI1 mRNA? I'm first going to kind of show you what our bell curves looks like. And thinking about IQ scores, I realize this is a very different than what I was just talking about. But whenever we're thinking about intellectual disability and our individuals with Fragile X, everyone is falling basically at the very end. They're usually not even on or even necessarily on this bell curve because they're below 3 standard deviations. And so the standard way of using these IQ tests aren't really that helpful or meaningful as a clinician or even especially as a parent. And so luckily there have been some methods at UC Davis that David Hessel has developed to really help extend that floor called the deviation IQ method. And so I will be talking about that a lot today because again, you can get basically a zero IQ to help us really expand what those individuals look like. So in a sample, this is our sample of individuals we have FMRP as well as IQ data on. So about 51 males and 29 females. And then I break down the males based upon whether they have no protein expression, trace protein expression, as well as our mosaic males. Not shockingly, our males with IQ are a lot lower than our females, but you still have a pretty decent range there, even within those males. And so, does it relate? Well, kind of. In our females, we have a pretty nice curve. We have, a 0.5 Pearson correlation. They're all hugging the line pretty well. So there is some evidence that, that FMRP levels are correlating or relating to those deviation IQ. So if you give me a protein level, I could give you an estimate of probably where their IQ is. On the males, it's a little bit different and it's not statistically significant, but what you notice is there's just a clump around 0 or a little bit, like less than 1 picomolars of protein. And so what I was thinking about as a clinical psychologist is, well, where are these falling? Is this a normal distribution or is this all just a mishmash of things? When you plot the histogram of this, it's actually kind of like a nice normal curve. And so this is really interesting and a little bit paradigm shifting of because it's sort of like, oh, they all have intellectual ability. They're all the same. But really, it's just a shifted IQ distribution in Fragile X, and at least among these males. And so, it can be really useful to think about maybe that an individual could have a normal IQ relative to other Fragile X males, or they have an above average IQ relative to other Fragile X males. That's something useful information you can give to parents. That's useful information as a clinician to know in terms of planning immediate behavioral interventions and even long-term planning. The females looks kind of normalish. It's not as beautiful, but again, sort of this idea that there might be some normal distribution here. We also have fewer females in this. And so if we are thinking about our second paradigm shift of how we're viewing Fragile X, is that these males, again, with trace or no protein expression, might have these normal distributed IQ that's downshifted 5 standard deviations, but otherwise relatively normal. And so we can start thinking about how each individual or each kind of subgroup of full mutation or females with Fragile X might have their own curves. Digging into the literature more, this finding is not unique to Fragile X at all. Prader-Willi syndrome has its own curve, Williams syndrome has its own curve, et cetera, et cetera. And so this is definitely maybe a new way to think about intellectual disability and single gene disorders in general, really thinking that maybe these individuals have their own standard curves. But the thing is kind of going back to my FMRP question. All right, so FMRP might downshift everything 5 standard deviations, but there's still a ton of variability in that cognition. What is leading to that? And I talked about this a little bit already, so I'm going to move on. So what accounts for the remaining variance in cognition? Well, probably relatively at some degree, all the things that we think about count for cognitive variance in our normal typical population. We have parental IQ, we have parental education levels, we have prenatal exposure, nutrition, SES, zip codes, all of the things. And so what this could potentially also relate to our cognitive variation in Fragile X as well. I also imagine and hopefully can figure out, is our CGD repeats and methylation rates that we think of specific to the Fragile X biology influencing that. And then lastly, I'm kind of adding the question of neural hyperexcitability as well, as that has been a very consistent finding across the mouse model slice physiology in our human individuals as well. And so, you know, if we can figure out, or if we can start unraveling what accounts for some of this additional variance, what can we do? Well, if we can target something that, you know, targets that hyperexcitability, can we actually move something from someone who is in that lower or below average normal for Fragile X to the above average rates? Are there things that we can do intervention-wise, pharmacological-wise, that can also push individuals to potentially better outcomes, better functional outcomes, and better quality of life? So let's talk about a little bit of clinical outcomes and what we're doing in our lab right now. So like I said, kind of thinking all of these things as potential mediators that are going to be influenced the way that FMRP impacts cognitive or other functional outcomes in Fragile X. And so right now we know that, again, hyperexcitability is really ramped up in Fragile X. And so how is that impacting cognitive outcome? So really quickly, just kind of showing some data across our in vivo slice models, our knockout mouse, and then our humans with Fragile X. And again, highlighting that gamma hyperexcitability, so that high frequency, sort of that background noise, so to speak, is really ramped up in Fragile X. We know this is related to our sensory hypersensitivity indicators by parent report measures. We've seen that this is related to some of our auditory attention measures as well. And so if we are able to target neural hyperexcitability with a pharmacological treatment or something else, would we then be able to later impact cognition? And so just looking at things kind of without drug input, we are seeing that it's not the greatest correlation, but there's some things kind of maybe starting to pan out. That if we're looking at FMRP across our males, females, and controls, that potentially those with lower FMRP tend to have higher of that gamma as well. And so in a post-baclofen acute dose, we do single dose trials where it's drug or placebo. They're doing the measures. 4 hours later, they're doing the post-dose measure, including EEG, some R bands, and some other computer cognitive-based testing. What do we see? So in a sample of 16 individuals with predominantly male, we're seeing that if you're just looking at males based upon whether they have protein, no protein, trace protein, or mosaicism, we're seeing that those males with no protein and trace protein expression are showing the most change in that gamma power. So gamma is reduced. That's what we think we want to do because we see it hyperexcitability at rest. So gamma is being reduced post-dose of baclofen. But let's think about this obviously with, again, that FMRP as a continuous variable. And so what we're seeing is that FMRP levels are significantly moderating this relationship between gamma change and drug, so that those individuals with the lowest levels of protein are having the greatest amount of change in gamma. So again, we're reducing that hyperexcitability in the brain. And so we're thinking with less gamma, perhaps there's less noise and they might be able to better key into their environment. Importantly, we're seeing that those individuals where that protein, or sorry, that are reducing in gamma also are showing that they're remembering more words in a list learning task, and they have fewer omission errors also during a task of attention. So potentially, again, this idea that if we're kind of reducing some of that noise in the brain, they're better able to kind of attend and be in their environment, we actually might be seeing more clinically meaningful change. It's not just the brain. If you went to one of my Gatlinburg talks, I did also show some data that, you know, it's potential, at least in the females, that we might actually be hurting their brain a little bit by giving them a change in gamma that actually could worsen some of their cognitive outcomes. And so it's really important to think about not just cool target engagement. We got the brain to reduce its hyperexcitability, but what does that actually mean? It seems like the females have potentially adapted. And we have some additional phenotyping data to show this too, that potentially the gamma is kind of a protective effect in the females. And so, you know, it's not going to be just one size fits all for Fragile X, especially given how much individual variability there is there.
Or real quick, that's just single dose.
This is a single dose. Yeah.
Okay.
Yep. And so, as I mentioned, we have some initial evidence that FMRP expression is related to these translational EEG biomarkers of hyperexcitability. In Fragile X, we think that FMRP might be mediating this underlying neurophysiology, which in turn might mediate that treatment response, and maybe even some of those cognitive functioning. We're collecting more of that baclofen single dose data now. I think we have 5 more participants so far, but hopefully getting at least 20 to 25 more. And so hopefully we're replicating and extending these findings. So kind of going back to my model, I mean, you know, in this perfect world, it would be really nice that if neurohyperexcitability really is kind of meeting this relationship between FMRP and cognitive function, and if we're able to, in those individuals with lower FMRP, we're able to lower that neurohyperexcitability, potentially we do have that promise of improving cognitive function. Obviously, we have a very small subset of data right now that is kind of showing that, but this is kind of a way to bring all of these pieces together to really think about the variable response to pharmacological intervention that really just hasn't been fully appreciated in Fragile X, despite anecdotal evidence from physicians and despite those known genetic differences of mosaicism sex in Fragile X. If you have been in the Fragile X world for a little bit, you know about the failed seaside therapeutics, where about 1/3 of individuals and families really thought there was a response there, but they considered it a failed trial and they got rid of it. Well, our group was reanalyzing that data and talking to some of those folks. They didn't even think about removing females from the analysis, or even just like looking at the different sexes differently. And so it's really important as we're thinking about some of these Fragile X trials moving forward, not only appreciating mosaicism and sex differences, but also then FMRP as a continuous variable as well, because it's very possible that these drug responses likely depend really on that underlying biology, and how FMRP at this point might be a really nice proxy for that. And so, like I said, we're doing a lot of reanalysis of our past data, looking at both FMRP as a continuous variable, if we have that on those individuals, as well as using mosaicism status at least as a binary variable as well. And then we can also think forward to how FMRP expression may be really important for those trial stratifications in the future to kind of really bring a step closer to precision medicine. And so, you know, if my own future directions, my own passion projects of this are really trying to figure out where is that variation in cognitive function. We really have, despite it being a single gene disorder, there's so much variation in the Fragile X population, even among those full mutation, fully methylated males, or what we're calling them fully methylated at this point. And so really kind of trying to uncover some of those biological and social environmental determinants of what's happening and how can we maybe better identify some of those facilitators and barriers to cognitive outcomes. And then in general, really just want to reduce those barriers to research and increasing generalizability of our studies. Right now, we have a very predominantly white sample of individuals with Fragile X that are coming in. These are also individuals who probably have lower than normal anxiety and irritability that are able to actually come into the lab and participate in our studies, put a cap on our head. And so my group just started trialing some home visits to be able to access families in their home, in their safe environment, in hopes of creating not only a more representative sample of Fragile X itself, but also reducing some of those additional barriers, whether it's virality, social SES, and race and ethnicity as well. And then, you know, lastly, continuing to examine kind of all of this FMRP and molecular data in relation to drug response. I'm really excited about that data and really think there's something there, and really think so much of the past trials that have quote unquote failed in humans might have failed in that larger subset, but whenever we reanalyze this data and look at a smaller, kind of more enriched subset, we probably are going to see some nice signal there. And so thank you for everyone that is part of the Cincinnati Fragile X Center. And everyone at Cincinnati Children's, but especially thanking for our families that have participated. And I included that picture with parents' permission of one of our first EEG visits. He had his best EEG capping and data collection at home. And so I'm really feeling pretty passionate about that's kind of the way our lab is going, or at least my lab is going to move forward. Yes, Matt.
This is great, first of all. It's really exciting stuff. Will say, I think we've talked about this as a field of those failed trials and how to actually pull out subsamples of individuals, because there were these anecdotal stories and some selective secondary analyses that showed there was some signal in there. So it's really exciting to see that somebody's actually doing this and making headway with it. So that's just fantastic to see. I have a few questions. I'm just going to start with the biology, though.
Yeah, try to answer.
Well, I'm just curious, I think more on your thoughts on relationships between CGG repeat length, protein, RNA, and the relationships that you're seeing. And so I think you probably know where I'm coming from in some ways on this, that the relationship between phenotype and between RNA and protein is so different in the pre-mutation range, right? So you, like, as you showed—
I'm not touching it.
right. Well, yes, we're happy to talk about ways to get to some answers on this story. But of course, the elevated RNA seems to be leading to toxicity in the pre-mutation, but protein levels are usually pretty normal. So you showed some data that RNA may be predictive, that protein may be predictive. So my real question is, A, how are you thinking about this? But also, have you looked at relationships between IQ and between gamma power and RNA versus protein? Is one a better predictor than the other?
Yeah, the permutation data, for the reasons you mentioned, the toxicity of mRNA is very complicated, and we're like, the lab has started doing that, and I would encourage you to look at our mRNA paper, because we have some wonky permutation findings that we didn't expect. And so I know we've talked or started talking to like Peter Todd's group and things of like, how can we conceptualize this? And I think there's just like another black box in the pre-mutation world that we have not fully delved into yet. Happy to talk more about that. In relation to your second question, I haven't looked at that yet. And I think it's a really good question, and sort of obviously an area of the things that I want to do in the next few years is really figure out, is there a combination of these variables in terms of FMRP, CGG, methylation, gamma hyperexcitability that could be sort of really predictive of things, because you would also imagine that the amount of FMRP is probably influencing how much hyperexcitability you have as well, potentially. And so I need, a friendly stats person at Cincinnati Children's that also kind of think of some of these path analyses of how it's all kind of mediating, moderating together. But I imagine it's going to end up being some complicated factor of that. And that's why I'm also thinking about a lot of those social, environmental, and SES factors as well, because even if, again, kind of referring back to some of our Gatlinburg talks, that we saw that pre-mutation mothers with higher education was sometimes a protective effect. And so you would also imagine those mothers or those individuals that have higher education, more enriched environments at home, are going to have different cognitive outcomes also compared to individuals that might come from lower education families or low SES families, because there's just not as many opportunities. And so it becomes it's a complicated biology picture. Then you add environmental stuff and all of that stuff. And yeah.
Good thing you're just studying one gene.
Correct, exactly. That's why single gene disorders are so easy.
I have a question. It's a fantastic talk that I also listened to a portion of when I had time at Gatlinburg for your other talk. So first question is, so in your paper for the diagnostic optimization analysis, you had mentioned, and also hear Peter Todd's name. So what was the relationship between random translation and as a potential confounder of some of the variability?
That is above my pay grade and biology knowledge. So, I'm gonna plead the fifth on that one.
Okay. And then knowing that in the diagnostic lab, so for detection of individuals with Fragile X, oftentimes it's already been done, it's not a panel, and there is a traditional route to say the individual is identified as having FXS.
Yeah.
So beyond that, exome sequencing is typically not done, panels are not done, and no knowing that there could be some kind of modifier effect for some of this maybe variability.
Yeah.
Do you have the samples in which you can run them, or have they done exome sequencing on some of these individuals?
Yeah, that's a great question. Clinically, yes, we're moving in that direction. And so if I can probably, let's see. Oh, I just shut out something. I was going to show my graph again, but okay, well, sorry. Yes, so if we're thinking about especially that IQ FMRP plot, for example, there were some individuals that like fall so far beyond off kind of an expected line. And so we're starting to actually kind of use that, or using our FMRP values to really think about who might have that like double hit and send them for additional genetic testing. Because at this point, one of the things that Craig and I joke about is like how we truly believe in our FMRP assay is we're kind of have like a little game of like predicting where they are going to be, how much protein expression is based upon their functioning and kind of clinical presentation. And so when individuals kind of fall out that way for one reason or the other, either they're higher than we expect or lower than we expect, we are thinking about some of those other genetic modifiers. And so let's see if I can go back to at least this slide. And so this individual grew up in a Romanian orphanage. And so you can imagine that there's some potential other influences, like very early life trauma that happened there. This individual we've sent for additional genetic testing and haven't gotten it back yet. And so, you know, we have been able to or at least think about those things. And all of these, 90% of the individuals that we're seeing for research are also in our clinic or relatively seen in our clinic. And so those are definitely things we're thinking about. The other really cool thing, and why I have my new fancy title of Director of Research Translation Engagement, is that we're starting to send our FMRP levels back to families. And so they're getting a letter whenever we're getting other levels and sending those individual values. Obviously, with the caveat that we don't really know too much right now about what it means clinically outcome-wise, but it's really important that when not only do we believe in our assay and that those numbers are real, but these families are participating in our studies, and we really want to make sure that we're giving them back some type of answer. And so.
What do you tell the families that are in that yellow circle in regards to the significance of protein levels, right? Because that yellow circle seems
really where the majority of our people are falling.
Well, the majority of the males, but also just that's where there's just this huge variability. It seems like there's some sort of moderating influences, at least on IQ broadly, right?
Yeah.
But that just seems like a more stochastic group. So if you're in this bin, we kind of don't know, but like,
yeah, that's exactly what we're telling them. And we have like a one quick pager of like, this is your level, we're still doing research, here are like our research papers about it. But then we have like 4 additional pages of, you know, we kind of expect people to fall in these general areas. Unfortunately, with that full mutation right now, it doesn't seem to be the only thing predicting, and there's a lot of that variance yet. We don't yet know where that is. And so it's an answer, but it's not a great answer for families.
Sure.
But hopefully I can get an RO1 to help figure that out.
Should be funded. Yeah.
You talk about mosaicism, and it sounds like usually I think of mosaicism in terms of like X chromosome silencing in females. That's like classically where I've heard that term before, but it sounds like you are talking about it as distinct from X chromosome silencing. Can you speak a little bit about that distinction and what mosaicism means in this context? And then I probably will have follow-up questions based on the answer.
That's so hard, Laura.
So mosaicism in males comes in 2 forms, like I said. And so you can have mosaicism within repeat counts. And so some of those some of those cells or X chromosomes are going to have a full 200 repeats, and then some are going to be in the permutation range or even in the normal range. And so some of those are going to produce protein and some of them might not. And then the methylation is somewhat similar in that not all of those are going to be fully methylated, and some of them are only going to be partially methylated. Does that answer at all?
Yes, it does. Okay. I'm sorry if I missed this earlier.
Oh, no.
So in like when you think about X chromosome silencing, you know that there are like, you know, clusters of cells in various places that have, you know, one or the other X chromosome expressed. Do you see that same sort of pattern in the mosaicism that you see in males that's driven by this?
No, to the best of my knowledge, that has not been figured out in Fragile X yet. I would say by and large, I feel like there's not a lot of work done specifically on the biology of mosaicism in Fragile X males. So I think it's a huge question.
Well, and I asked the question because I saw a talk by Huda Zoghbi several years ago where she was kind of teasing apart the like X chromosome silencing females with rats, and essentially demonstrated that, you know, depending on which cells are expressing the impacted X chromosome and how that is distributed in the brain, like you're gonna get a phenotype that is different, that may be more extreme if you have more cells in the brain that are, you know, expressing that affected chromosome, and then where in the brain impacts it as well, which like makes a lot of sense. And explains a lot of the variants that we see in behavior and Rett syndrome. And so I'm wondering if the same kinds of things can be playing a role in Fragile X as well, especially in that male phenotype where obviously we haven't explored it quite the same way.
Yeah, I mean, they're definitely thinking about at least the X inactivation in females in a very similar way. And, you know, all of this is peripheral blood. And so we're just making the general assumption that it's somewhat similar in the brain, but not only are you making the assumption that whatever we're getting from, you know, your arm is similar to what the protein production you have in your brain, but then you also have to think about what areas of the brain are you having that? And are there little pockets in frontal cortex versus auditory cortex and all that? And so it gets a very even more complicated picture. I know some of our colleagues at UT Southwestern has developed like this 15% FMRP mouse. Unfortunately, it's like a little bit of a different biology than what the human experience or what the human biology looks like, but they are gonna, I think, start doing some brain slices just to at least confirm that it all is sort of similar in the brain, but again, it's made differently than how the human biology works. So I don't know how helpful that actually is gonna end up being, and I don't think there's enough postmortem tissue data out there for the Fragile X world to really start diving into that.
Is there, I'm thinking the relationship between FMRP and IQ that was in the females. I mean, is there variation in FMRP expression in just typically developing controls? And then is that same relationship holding in people without Fragile X?
So the relationship in typically developing controls isn't really much one between FMRP and IQ in typically developing individuals. And so it does sort of seem like once you get to probably your average range of FMRP, it's sort of, you know, all of the other variants, all the other things. And so I would imagine that would similarly play out in females. I'm trying to find what graph I'm trying to find right now. And so does that answer your question?
It does. Yeah,
okay.
So it sounds like you mentioned terms like you just have to hit a certain point and then be
and I would imagine that is similar to in males too, is that right now we're what we are defining as trace expression males is based on a product of the lower limit of detection of our assay. And so it's possible that even lower levels are there that we're not detecting that could be meaningful, but it also could be possible that you need a specific amount of protein for it actually to be effective. And maybe it's not our IQ measure that's really showing that, but maybe it is one of our brain-based measures, or maybe it's adaptive function. And so that's definitely something like it would be really nice to be able to figure out, like, is it at 0.25 picomolars or whatever it is, like that's the number you hit, and that's the amount of protein you need to be clinically meaningful to really have any downward effect. But we obviously don't know yet, and just sort of.
So, you ready for a psychologist question?
Yeah.
It's flabbergasting. You have to come back down. How much people rely on IQ? To what degree do the subscales of IQ cohere?
$1 million question as well. Right now, that is mostly all abbreviated IQ at this point, and I haven't teased things apart yet. And so those are, yeah, so it's like a really broad estimate. And so with the initial U54 grant, we were doing abbreviated IQ on everyone, and which is a gross estimate, and for time reasons, blah, blah, blah. But we are moving in the direction of now collecting the full Stanford Binet on everyone to tease some of that apart to figure out, is there a specific subtest or a specific nonverbal something or other that maybe that's the thing that is kind of best estimate of relation to molecular stuff, but then also our functional outcomes. Because I don't think in general in Fragile X, they've really been able to figure out like, what's that maybe one area of cognition that can best determine broad functional outcomes or quality of life yet.
Right. And it's abbreviated is that, and that is, I mean, I completely understand you're doing so much other stuff that you're not getting a full scale, a true full scale. But even then, even knowing what the coherence is, and comparing that to the coherence in typically developing, is that okay to say this week?
This week,
typically developing populations, that it's just really it could be instructive in terms of, because we often find in developmentally delayed populations that tests of global intelligence or global, they're not nearly as sensitive, especially to interventions.
Yeah.
As for example, something like pulling out a can tab.
Yeah.
Or some other specific function.
Yeah. No, I mean, and I think, hopefully again in this R1 that I need to write, there will be kind of more thought around some of those specific cognitive tasks that we're using, and if there is something that's going to be one more representative of their true cognitive function, but then also, for broad scales. The graphs don't look as pretty for our Vineland. I've only started looking at that, but that's also because you shrug your shoulders, hard to figure out.
Yeah.
And so
it's surprising to me that a more granular assessment of cognitive function isn't part of the phenotyping. A lot of disorders, but that's okay.
I mean, I think a lot of that comes down to the fact that our IQ measures have been developed for a normally distributed population, not
and developed in some ways. Well, they don't even do a good job there, but developed to detect gross deviations.
Yeah, right. It's like, oh, you're impaired, so you're going to go in the bin, as opposed to trying to figure out like, what level of impairment are you at, and what maybe do you have strengths in relative to other areas of weakness. And I think that that was a topic certainly at Gallenberg this week of trying to really more granularly determine cognitive level in the population.
So it seems like you're using IQ because our understanding of FMRP is that is most profoundly probably influencing cognition. But it seems like do you think that there are also ways of modulating like sensory hypersensitivity, which I think seems to be related to gamma, right? Where you can sort of modify some of these other factors that then may also contribute to cognition. I guess my overarching question is like, were you looking at IQ because that seems to be most closely related to what you think is a downstream effect of FMRP, or were you looking at IQ because that seems to be the primary thing that you should be targeting to kind of improve everything else? That makes sense?
Yeah, no, I understand your question. I hid this slide, but this kind of answers your question or expands upon your question, then I'll go back to your question. So we have looked at FMRP in relation to other kind of core phenotypes of Fragile X. And so on the male side, it looks like lower FMRP is related to more stereotyped behaviors and higher levels of irritability. And then very interestingly, on the female side, again, these the ends are pretty small in this, just kind of putting this together. But on the female side of things, it's higher FMRP values are related to some of those psychiatric features. So more anxiety, more depression. And so we are seeing that, you know, FMRP, it's not just IQ that things are relating to. There are these other factors of Fragile X phenotype. But obviously the female side of things is kind of interesting.
Yeah, this is such an interesting.
And kind of thinking it maybe more along the lines of what we traditionally call like higher functioning autism, where they do have higher IQ, they are more self-aware, they're kind of more involved in daily life, kind of school and bullying and all of those things, they're more aware of those things. And so it's possible, I guess it's kind of one way I'm conceptualizing it. Also, you could then potentially have some of that toxic mRNA effects also within the female biology. And so it's also possible that there's just underlying risk factors in addition.
It's kind of interesting in how you think of like in autism, you see kind of different symptom profiles in males and females as well, where in females you see more of those kind of internalizing, you know, anxiety, depression kinds of symptoms, you know, compared to males, and this lines up really nicely with that as well.
Mm-hmm. I don't know if that fully answered your question, Catherine, or not, but it made me think of the data slide that I didn't show.
Yeah, I think that's kind of what I was thinking. I'm just sort of curious about like kind of the overall.
Yeah, I mean, what the primary reason that we initially looked at IQ is it is the #1 cause of intellectual disability. It's a primary phenotype, and so there's 5 or 6 past papers done over the past 20 years using different FMRP assays that have related FMRP to cognition in a predominantly female and mosaic male sample. And because they didn't have necessarily that sensitivity within the males to detect. And so, but we are, based upon kind of initial look at some of our other phenotypes within Fragile X, you would sort of imagine that FMRP is going to be related to those other core features, but obviously females potentially need to think about things a little bit differently and how FMRP can both have, a positive effect on cognition, but then you're increasing potential risk for some of these psychiatric symptoms, which is also important for, families to know. But clinically, I kind of would probably predict that anyway. If you're having higher IQ, the things that I'm referring those families to are going to be a little bit different than the strategies that I'm referring lower IQ females to.
I just want to keep monitoring the chat here as well for some of our virtual attendees. We have a comment about how the different phenotypes could also be related to different brain circuits for anxiety versus repetitive behaviors, etc. And then a question from Dr. Mohammed Raza. His question is about the level of methylation with the protein expression and mRNA levels. Is there any correlation of methylation of CGG repeats with the variable FMR1 in males and females?
Not yet, because right now our reports back from Rush just say fully methylated, and they don't give us any numbers or quantitative. And so with hopefully our new technology we have, we'll be able to actually quantify that better. So is it 95%, 97%, some 82%? And be able to look at that with FMR1 and the protein levels. So stay tuned.
Real quick, Lauren,
yeah.
Then we'll close out. I hear what you're saying about your next R1, but I feel like there's 3 R1s here. And so I did just want to check in. Are there plans for longer term studies of baclofen based on protein levels, sex, and gamma power?
That's our current project. It's one of our human projects right now. Yes.
Is that right?
Yeah. So
chronic treatment.
Not chronic treatment. Not yet. No, it's still single dose. We're trying to replicate our findings before then we would potentially do a chronic, acute versus chronic. So not yet. At least in the mouse, the chronic treatment looks a little bit better, not like substantially better, but
Yeah.
you would imagine that at least would hold the effect then. So yes.
Cool. Seems like there's also a developmental question in there too.
There's a ton of developmental questions there. And so I'm sort of the 8 and older data,
Right.
and then colleagues that are dealing with the 8 and younger data. And so, yeah, there's a lot of developmental questions out there that hopefully we'll get some papers out soon. That just data has been a little bit more messy and collecting longitudinal and pandemic hit, blah, blah, blah. So
Yeah. Those are going to be 3 great RO1.
I agree.
In the next year. Yeah, yeah. Thanks again for a wonderful
Yeah,
talk.
well,
Laura.
thanks for letting me come.